Okay, so as I mentioned, I would like to tell you a little bit about the rules that we're using to guide our medicinal chemistry. And I'd like to start with the reason why we have these rules. 20 years ago, we would basically synthesize a molecule, test it, see if it works or not, synthesize the next one. And that was not a very efficient process. And as you can see, the cost of developing a new drug is extremely high. There are different ways of calculating this, but one simple way is to take the number of drugs. Oh. And this one doesn't work either. Gosh, we have little problems here. It doesn't work. Yeah, it has no. It has. I think the battery is dead.
So maybe you can just tell me the next slide and I will be switching the slide.
No, he's looking for the pointer, Alexei. Alexei, pointer, not slide. He's got batteries, he's prepared.
Yeah, I don't know in which direction they go, so. The other way.
There is a pointer, and if you want, you can tell me to switch to the next slide and I can do this for you.
Yeah. So we can try. I prefer to admit.
The laser pointer is working.
The laser pointer is working? Okay.
And if you have promised to switch the slides, I can do this for you.
Thank you. Yeah, I don't know what's wrong with this.
Yves.
Let me see. Yeah, I think the batteries of this one are dead. Alexei, I think the batteries here are dead, that's why, yeah. So, okay, so excuse us for this. So, can you go back to the previous slide? Thank you. So there are, as I said, there are many ways of calculating what a drug costs to bring to the market. And one simple way is to simply take the number of drugs that have been approved by different companies and divide by the cost that has been invested in research to bring these drugs to the market. And as you can see, if you take the first example with AstraZeneca, they brought 5 drugs to the market, invested $58 billion between 1997 and 2011. If you divide this by 5, that gives you the cost per drug, which is about $12 billion. So it's a huge amount of money. And if you take even the more efficient companies like Amgen, for instance, which has produced 9 drugs at the cost of $33 billion, it's still $3.7 billion. So it's really, it's very expensive, it's very difficult, very long to bring drugs to the market. Next slide. So if we can do this a little better, of course, that would help tremendously. Of course, it's not as dramatic as it seems, because if you can find a good drug like AstraZeneca did, for instance, with Nexium, you can sell it worldwide and actually recoup some of the money that you have invested. But still, not all drugs bring so much money back in. And on the next slide, in particular, many of the drugs that we try to develop fail. So the question is, for us chemists, can we do it better? Can we do it cheaper? And can we do it faster? So can we select drugs better, drug candidates better, before we bring them into clinical development? And that was the motivation for developing rules for drug likeness. The strategy was simple. It was to define the properties of drugs that reach the market, that reach the patients, and develop candidates that would have similar properties overall. So the expectation was that these property-based rules would help the medicinal chemist in 3 main different ways. The first one is to be predictive and allow to synthesize only the most promising candidates. Then it would give us a way of keeping track of the numerous properties that are followed in the multiple parameter optimization effort that is drug development, and then ultimately enhance the probability of success by limiting parameters that are difficult to predict, like toxicity or bad pharmacokinetics. Next slide, please. Yeah. So that led to the development of a number of rules, and the first one was developed by Chris Lipinski, which is known now as the Rule of 5. And basically what it says is that after studying 2,200 drugs, one thing, it appears that those that have a good oral absorption and a good permeability have properties that tend to be below a molecular weight of 500, a lipophilicity measured as C log P below 5, less than 5 hydrogen bond donors, and less than 10 hydrogen bond acceptors. So that was in 1997, the first rule. Soon thereafter, in 1999, thank you, Gose published a refined analysis with looking at 6,400 drugs or drug candidates and defined criteria that would characterize most of them, actually more than 80% of the established drugs. So not only looking at oral absorption and permeability, but looking at the overall properties of drugs. And the result of this analysis is that if you look at all drugs, their log P is between minus 0.4 to plus 5.6. It's a relatively broad spectrum. We looked at molar refractivity, molecular weight, atom count, etc. And if you categorize these drugs in different groups, for instance, antidepressant, this range decreases a little bit. So for instance, log P between 1.4 and 4.9, molecular weight between 210 and 380. So that gives some guidelines of where the properties are that would be the most favorable for certain classes of drugs. Next slide. The problem is that actually it's not just the quality of a drug that makes it successful. It also has to do a lot with the target that we're trying to modulate. And if you think of the physicochemical properties and look at how they depend in function of their target, you see that simple parameters like the molecular weight is smallest for ion channels. It increases for aminergic GPCRs, kinases, etc. Lipophilicity follows the same trend, being the lowest for ion channels, proteases, and then increasing for kinases, aminergic GPCRs, transporters, peptidergic GPCRs, and nuclear receptors. So all of this, as a conclusion, really, the whole process is complicated. It is not just chemistry, it is also biology. So we need to understand a little bit what are the overall parameters that influence all this. Next slide, please. But these guidelines, as I said, are important, and I want to illustrate why they are so important. It's kind of obvious that if you think of it, a drug must have certain parameters to be orally active, to be sufficiently efficient, to be selective, etc. But this depends on parameters that are a little less obvious. And for instance, lipophilicity is one of them. We know that if you look at the range of lipophilicities, the best drugs or the best drug candidates are roughly between 1 and 3. And the reason is relatively simple. If you go to values that are below 1, you end up with molecules that have a lot of renal clearance, they have a poor permeability, they have properties that are overall not favorable to oral absorption. So they are not very good drugs. If you go to higher lipophilicities beyond a value of 3, then you get molecules that are much less selective, that distribute very much in the body, but sometimes also accumulate, that have a higher tendency to have unfavorable pharmacokinetic properties, and that tend to be toxic. So clearly this is maybe just a simple observation, but at the end of the day, a very important parameter to understand what are the limits of these different values that are acceptable for drugs. Next slide. And this ultimately culminated in the development of a model that is called the multiple parameter optimization model that has been published by Pfizer, which not just said a parameter must be, for instance, like the molecular weight below 500, but tried to refine this a little bit by associating every parameter with a function that could be simply a decreasing function, or that could be a hum function, for instance. And if you think of lipophilicity, that would be an example of a parameter that follows a hum function. Being too low, it's not good. Then it gets better. It reaches an ideal range. And then this, when the log P, the lipophilicity gets higher, the quality of the drug gets lower. So if you associate with each parameter for a drug a value that would be 1 at the maximum and 0 at the minimum, and maybe 0.5 at this point, then you can, for each parameter, give a ranking, and add for these 6 most important parameters the different rankings, giving you a maximal value of 6. So a compound that would not fulfill any of these parameters, that would always be here or there, would have 0. A compound that would always be at the maximum would have a value of 6. And next slide. And if you it kind of all makes sense. There's a logic behind it, but there's also the observation that if you look at all drugs, which are here, it's hard to see, but it's in black, and drug candidates in blue, the multiple parameter optimization score tends to be in the higher values for successful drugs. There are a few that have lower values, between 1 and 2, 2 and 3, but mostly it starts at values that are between 3 and 4 and higher. To the point that, if you can click once more, Pfizer decided to focus exclusively on developing drugs that have MPO value that is above 4, giving them, they thought, the best chances of success. Next slide. And of course, you can always do better. You can always parameterize this even more. And that went to the point that a parameter has been published to try to quantify the chemical beauty of a drug, trying to take all possible parameters into account, including toxicity, including physicochemical properties, aromaticity, etc., to give what is maybe a single value of the chemical beauty of a drug. Practically, it's of limited applicability because it really only allows comparison within one series, and I wouldn't really consider using it in a systematic manner, but it's an attempt to avoid making judgments that are too black and white and try to introduce a smoother comparator in this comparison. So, next slide. What we need to remember, however, is that all these guidelines are guidelines. They're not absolute rules, and they have limitations. For instance, I mentioned briefly that the parameters depend in function of the objective of the drug, of the class. And you can see here, if you take a simple parameter like the molecular weight, for all drugs on the average is 338. Now, if you look at antibacterials for gram-positive bacteria, the average is 813, so it's more than double. For antibacterials that are for gram-negative bacteria, it's 414, so it's low again. So we need to understand what are the limitations and when to apply these particular parameters. If you can click. And of course, we need to deal with natural products, peptides, and biologics separately. They have different ways of interacting with membranes, with biological systems. For instance, if you look at cyclosporine, if you would have used the Pfizer multiple parameter optimization method, you would never have developed this drug. However, this is a fantastic drug that made the whole area of organ transplantation possible, highlighting the fact that we really need to understand how and when to use these parameters. Next. You need to consider the impact of alternative modes of administration, because of course what we see here is for orally available drugs, what we discussed before. Now, if you can apply your drug with a cream, or if you can give an IV injection for certain indications is perfectly acceptable, then obviously that will change all these parameters. And at the end of the day, what is really important is not to take this too seriously, only as guideline, to leave space for creativity and rule-breaking discoveries. Next slide. Thank you. So in conclusion for this introduction, it's really important to know these guidelines if you want to develop drugs that have a chance to be successful and reach the patient. However, you need also to understand when to follow them and recognize when to think creatively and once in a while break the rules. And this is my introduction to the presentation I want to do to you on this AMPA kinase antagonist that I was talking about for the treatment of epilepsy, BGG492. Next slide. Before I talk about the molecule, I would like to talk a little bit about epilepsy. Epilepsy is a disease that has been with us since the beginning of humanity. And we have writings, at least in this part of the world, that date back to 400 BC with the writing from Hippocrates, who recognized that the seizures were a disease of the brain. And it was a fantastic observation. It seems simple, but it took a very long time for for the world to understand that epilepsy is not due to evil spirits. Up to the point that, if you can click, we see in areas of the world that have lost this knowledge, unfortunately, appearing as late as 1494. A book like the Malleus Maleficarum, which is a textbook on witch hunting and which actually describes epilepsy patients as witches. Of course, it's very impressive to see a full seizure of an epileptic patient, and people who did not understand this thought that this had to do with evil spirits and with the devil. And unfortunately, that sent a lot of patients to their death, because during the Middle Ages, as you know, witches were burnt. Yeah. It took a while for this to improve. In the 16th, 17th century, a little more reason and scientific reasoning came back to the world in terms of understanding epilepsy, leading to the first drug for the treatment of epilepsy, phenobarbital, which was brought to the market in 1912. That was a revolution. A revolution because that really, for the first time, allowed to really treat patients that badly needed it, actually. And after this, next slide. The scientific understanding of epilepsy increased rapidly with, in particular, the development of the first electroencephalographic machine by Hans Berger in 1926 at the Jena University in Germany. And all this led to the development of new molecules for the treatment of epilepsy. There are currently about 20 anti-epileptic drugs that are available on the market. And I will tell you a little more about these drugs, and in particular about why we need more in the next slides. Just a word about epilepsy in Russia. The prevalence in Russia is 0.34%, which brings the total to about 500,000 patients in Russia. It's actually a little less than in the rest of the world. The majority of the patients with epilepsy are treated in Russia, which is great because it's still not the case in all countries. The main cause is head injury. 28% of patients become epileptic after head injury, or cerebrovascular disorders, 12%, and there are a number of other causes. The epilepsy is most frequent in men, slightly more frequent than in women, with a ratio of 3 to 2, and there's also a geographic parameter element. We see that the eastern population in Russia is more affected also by a factor of 3 to 2 than the western population. I want to just say a few more words about the causes of epilepsy by talking about three well-known patients. One is Peter the Great. Peter the Great was not epileptic when he was a young man. He actually became epileptic as the consequence of an infection. When he was 21, he got a very severe encephalitis, from which he recovered. He was a healthy and strong man, recovered from this encephalitis. Not everybody does. However, that left with him damage to his brain, which translated into, at the beginning, relatively mild crises of epilepsy, and they became, with time, they became worse and worse, and he remained epileptic until his death. The next patient, Dostoevsky, has written a lot about epilepsy, actually, indirectly, by including in his stories, in his books, patients that were epileptic. And it's very interesting descriptions of epilepsy. The causes for him were, they're not really fully understood. They were probably of genetic origin. And the third patient illustrates the third, one of the main causes of epilepsy, traumatic brain injury. Lenin was not epileptic until 1912, when he suffered, when he was the victim of this assassination attempt, which left a bullet in his brain, a bullet that could not be surgically removed. And the combination of the damage to his brain and the presence of this bullet caused epilepsy, which became worse and worse with time, and actually Lenin died in a crisis of epilepsy, status epilepticus, which is basically an extreme crisis of epilepsy which doesn't abate anymore and leads to death after a relatively short period of time. In the world, there are 50 million patients. 70% of the treated patients are seizure-free. What is interesting and what fascinated me since the beginning is that 30% of patients are completely resistant to all treatments. Those that are treated and become seizure-free can have an almost normal life. They slightly suffer from the side effects of the medication, but this is nothing as compared to these 30% of patients that cannot be treated at all. And some of these patients try 5, 10, 20 anti-epileptic drugs and remain subject to seizures all their life. The reason is that from the 20 anti-epileptic drugs that I mentioned before, most drugs act via GABA-A agonism or the blockade of sodium and calcium channels. So it's basically two mechanisms. 20 drugs, only two mechanisms. And there is only one that is based on the direct antagonism of glutamatergic neurotransmission. It's perampanel. It's a non-competitive AMPA antagonist. A compound that has its problems. It's not fully effective and it has side effects, but it is the first one actually that is really acting via a very different mechanism of action. And why do I highlight this compound? Next slide, please. I highlight this compound because the glutamate receptor, which is the target of perampanel, is the glutamate receptors are the main neurotransmitter in the human brain. And actually the glutamate acts on two types of receptors, the ionotropic receptors, which are ion channels and responsible for the fast neurotransmission in the brain, and metabotropic receptors, which have a more modulatory mode of action. The ionotropic receptors are directly linked to epilepsy, and actually their overactivation leads to epilepsy very rapidly. The main culprit is the NMDA receptor, and we know that blocking the NMDA receptor and transmission of calcium through the NMDA receptor prevents epilepsy. The problem is that we cannot block NMDA receptors. They play such a fundamental role in the functioning of the brain that if you decrease the activity of the NMDA receptor, it comes with a lot of side effects, and they are just clinically unacceptable. One option, however, is to try to block the AMPA and kainate receptors, which indirectly modulate the activity of the NMDA receptor. And this is what we've tried to do, to find a drug that would act on both AMPA and kainate receptor, in contrast to perampanel, which only blocks indirectly the AMPA receptor. Next slide, please. And we've done this with developing two drugs. I will go very quickly through the first one, which is MP397, a competitive AMPA kainate antagonist, our first clinical candidate, which was developed starting from this quinoxalindione here, CNQX, which has been published by Théhonoré and in Science in 1988. This compound was the first one to act on the AMPA receptor. It's fairly selective. So it's a very good pharmacological tool. However, it is so extremely insoluble that it absolutely doesn't work as a drug candidate. It's orally completely inactive, for instance. We therefore try to modify CNQX by introducing a side chain that would have a nitrogen to make a salt, make it more soluble. And actually we succeeded by introducing modifications in this region, going from a compound that was relatively weak, 800 nanomolar, to one that was a little more potent, 300 nanomolar on the AMPA receptor. This compound is tenfold more soluble now, but it is still inactive after oral administration. The problem, we believe, is that we started from a compound that is really like a stone. It's absolutely insoluble. This one is 10 times better, but it still is not soluble enough and doesn't dissolve in the stomach. So we made further modifications, and we introduced a very strongly solubilizing agent, which is a phosphate. Phosphate is what you find in suds. When you do your laundry, you're using phosphates. And basically the idea was to introduce soap in this molecule to make it water soluble. And actually that worked very well. This compound now is more than 1,000 times more soluble than the starting lead. It has this aminophosphonate, which is the basis of this solubility, and which also increased the affinity for the AMPA receptor to an IC50 of 14 nanomolar. So a compound that would be about 80 times more potent than CNQX. It's highly water soluble, as I mentioned, and in the electroshock seizure assay in mice, it has an affinity, it has an efficacy that leads to an ED50 of 5 milligram per kilo after oral administration. So that's the drug that we wanted to find. And I'll show you with the next slide how it works in patients. So you have to remember that our objective was to go for patients that are treatment resistant. And there is a category of patient that is treatment resistant and that is so desperate that they go for surgery. So basically they go for a surgery that will remove the part of their brain that causes the epilepsy. It's pretty extreme. And these patients are really, as I mentioned, desperate patients. They have tried, we had one of our patients here that had tried 18 anti-epileptic drugs, basically all of them. Still had seizures and couldn't live a normal life, by far not. So before doing this surgery, the physicians need to basically define very precisely where is the area of the brain that causes the epilepsy, to remove just the strict minimum. And to do this, they need to observe epileptic crisis. So they remove the drugs from the patient, so they take the patient away from the drugs, and these patients go into seizures relatively quickly. And during this period of time, we can try to give them our drug and see if it works. If it works, they don't have seizures anymore. If it doesn't work, the physician gets the information he needs to do the surgery anyway, so there's no harm to the patient. And you see here the results of two cohorts. In red, these were the patients that were given a placebo, and you see that very quickly they had enough seizures that the physician could say, stop, now we have what we need, we can do the surgery and take this patient out of the clinical trial. On the blue line, we have the results that were obtained with becampanel, AMP397. And as you see, we lost a patient on the very first day, the second day, and then none anymore. So it's a fantastic effect, actually. It's a fantastic effect when you think that these patients are so extremely treatment resistant. So we were really very happy with that. And continue developing becampanel until, the next slide, until happened something that I will talk about after just coming back to the discussion that I started with. Did we follow the rules? We started with CNQX, and as you remember, we had these rules, this ideal profile that was defined before that showed what a drug should look like. And if you click once more, we see, can you click please? Yeah, thank you. We see the profile of CNQX, the lead that we started from. We see that this compound is way too hydrophilic. It has a polar surface area which is very high, 3 times more than what we started with. That's what it was not such a good drug. Now AMP397, to our surprise, or actually to our surprise, to maybe the surprise of those who developed the rules, has a profile that is even worse. All these parameters do not fulfill the requirements at all. So clearly that's an example that in some cases you need to look for the exception. And AMP397 is an exception. It's a molecule that worked very well in patients that, however, doesn't have the parameters we were looking for at all. However, you might say, is AMP397 a drug? Maybe not, because we didn't make it all the way. We had problems, and the problems are illustrated on the next slide. They could not be predicted by the rules, but AMP397 actually binds to bones, and it is orange. So after a while, the bones turn orange, bright orange. And we didn't know this, of course, we didn't observe this in patients, but in the animal species in which we did the tox study, after 1 year, we could see that their bones had turned bright orange. And we considered this as unacceptable, and we decided to look for another molecule that would have the same properties as AMP397, so be a strong competitive AMPA kinase antagonist, as efficacious in vivo as AMP397, but without binding to bones, and of course being colorless ideally to avoid any potential problem linked to color. So the question was, are we going to try to synthesize another quinoxalindione? And we knew at that time that it was difficult. Quinoxalindiones are not very soluble. They have these very extreme properties, and we thought, let's try to go away from that. Let's try to see if we can find another chemotype that would have properties more drug-like than AMP397 was. And we did this with the next slide, please. Starting from the observation that the quinoxaline dions, which we tend to draw this way, are actually partially deprotonated in vivo, and they actually look like this rather. And the question is, can we mimic this form of the quinoxaline dion, which is actually what allows the quinoxaline dion to mimic glutamate and bind to its receptor. And we first started with cyclic hydroxamic acids, which have a hydroxyl group of a similar pKa in this position. And actually, the idea worked. Unfortunately, these compounds were only moderately potent as competitive AMPA kinase antagonists, and in vivo they didn't really have any strong activity. So we worked further, and we thought that one of the reasons why this compound was so poorly bioavailable and so weak in vivo was that the charge is very localized. And the chemist who worked on this, Emmanuel Koller, had the idea that if we could replace this hydroxy group by a bioisosteric and sulfonamide of a similar acidity but with a more extended charge distribution, these compounds would become more bioavailable. And actually this is exactly what happened. He replaced the OH group with this methyl sulfonamide. The pKa 6.9 is very similar to what we had before. And even though the first compound he made was quite weak at the AMPA receptor, 2.4 micromolar, the activity after oral administration is fantastically good. If you compare with the MP397, we had an ED50 of 5 milligram per kilo, so it was 4 times more potent. But in terms of affinity for the receptor, it was 200 times more potent. So indicating that these compounds are very bioavailable. Next slide. So we had the lead. Now the problem was, as I mentioned, that the lead was relatively weak. And we decided to try to use the structure-activity relationship we knew of the quinoxaline dions to combine, make a hybrid with our new orally bioavailable compounds. And that gave us molecules like this one, which is the exact hybrid of these two, which we thought would have both the affinity and the oral bioavailability. Unfortunately, it doesn't always work as we want, and in fact, we combined the bad properties of these two compounds with the molecule that had a relatively weak affinity with the 6.5 micromolar and no oral activity whatsoever. So we thought, yeah, maybe we've been exaggerating a little bit. We knew that here the polar surface area was way beyond what the rules would predict, we had found an exception, but maybe not all compound can be an exception, and with the polar surface area of 220 square angstrom, we are probably way too far. So we tried to decrease this parameter. And we started again making hybrids, starting from, for instance, YM90K, which is a compound from Yamanouchi, which has an IC50 of 89 nanomolar and which is less hydrophilic. It has a polar surface area of 132 compared to 180 for MP397. We combined it with our lead, we obtained this molecule, which we were happy to see has an affinity that remains in at the same level as YM90K. So we had a good affinity, we had a polar surface area which was, yes, too high, but a little better than the one we had obtained before. And we started showing the first sign of activity. Even though this compound was not orally active or weakly, it had a good activity after IP administration. So with this, we decided to continue to explore this series. And on the next slide, you see the challenge we had to struggle with. The problem is that in this series, we have to mimic glutamate. Glutamate is a very small but very polar molecule with a C log P of minus 2.7, very hydrophilic. And our first mimic of glutamate, MP397, was also a very hydrophilic compound with also a very high polar surface area. So these are parameters that are contrary to oral bioavailability. So we had to find a way of optimizing this a little more. Next slide. And yeah, again, the requirements for binding affinity are such that when we have a high affinity, we have a low oral activity, and as a consequence, we had to find a way of combining these two parameters, finding a compromise. And to do this, we have used two techniques that are illustrated on the next slide. One was using co-crystallization of our drug candidates with the receptor to try to understand how to maintain and maybe improve the affinity, and that worked actually very well, very quickly. We could improve the affinity with a molecule like this one, which is an IC50 of 12 nanomolar. Of course, no oral activity due to this very strong polar surface area. But then the second thing we did was to change the way we run our optimization. We first did the synthesis of the clinical candidates. We looked at the IC50, the affinity for the AMPA receptor. And when this was good, we directly went into a cassette pharmacokinetic experiment to determine the bioavailability. If the bioavailability would not be good, we would not characterize these compounds any further, go back to the design and the synthesis of the next candidates. Now, when these compounds would have enough affinity and a good enough oral bioavailability, then we would test it in the mouse electroshock-induced test in mice. And that is what allowed us to identify our next clinical candidate, BGG492, this molecule here, which has a little less affinity than we would like to have, maybe, but again, this is a compromise with an IC50 of 190 nanomolar and an efficacy in the mouse electroshock model of an ED50 of 6 milligram per kilo PO. So you remember AMP397 had an ED50 of 5 milligram per kilo after oral administration. So basically the same range of activity, even though the affinity was weaker. So we had found a compound that had, by modifying its parameter, a better oral bioavailability. Next slide, please. Let me say just a few words about the in vitro pharmacology of BGG492. This compound is active on both the rat and the human receptor. It's important to be able to predict the activity in human. The functional efficacy was determined by electrophysiology. In the rat cortical wedge, we see that the affinity at the AMPA receptor is the same as at the kinase receptor, the second receptor we wanted to inhibit. However, it has no activity directly at the NMDA receptor, which is good because the NMDA receptor, as we know, is the one that causes the side effects. And we characterized this compound in a panel of another 150 targets. There was no activity whatsoever for any of these compounds. So any of these targets, a very selective compound at the end of the day. Next slide. I want to go very quickly through the pharmacokinetic properties, which is an important parameter to understand when you optimize a drug, because the parameters are important to understand the activity in vivo. If you can click once, please. Alexei, just once? Yeah, thank you. There are 3 points I would like to make. The first one is that the exposure, the oral bioavailability is very high in mice, as you can see here from the plasma exposure. It's also very high in dogs, but it's a lot weaker in rats. And the consequence is that in our rat animal models, we will have less efficacy. However, in man, which is usually, in terms of oral bioavailability, very similar to dogs, we expect to have a high exposure. Next one. The second parameter that is important to know about is the duration of action. And we see that it is similar, 3 hours roughly, in mice and big old dogs, expressed in terms of half-life. It is also, again, faster eliminated in rats. So we will see a short duration of action in rats. Important parameters to understand, to compare the different models. And the next one is the brain to plasma ratio. It was a surprise because this compound looks like there's only 3% or 2% of the compound that penetrates the brain, which is very weak. And actually it looks like contamination from the blood. And in fact, we had to do additional experiments to convince ourselves that the compound was really penetrating into the brain. And it does. It penetrates into the brain very well, in fact, but it doesn't penetrate into the cells of the brain, which means that when we do a pharmacokinetic experiment and homogenize the brain to look at the concentration, we dilute the compound and we don't see its real concentration. But as you can see on the next slide, the compound does penetrate the brain actually quite well. This is an experiment that is done with rats, an electrophysiology experiment with rats that are implanted with multi-barrel electrodes that are used for two purposes. One is to measure the activity of the neuron, and the second one is to use the electrode to inject AMPA directly into the brain at the point where we measure the activity, and AMPA is the natural, is the agonist of the AMPA receptor. And we see that when we do over 1 hour multiple measurements, multiple excitation, we get about 100% response during the first hour. It varies a little bit, but not very much over this course of time. And then during the second hour, which is this curve, after a while we apply BGG492 in the periphery, so not in the brain. It takes a little bit of time for the drug to distribute in the body, to penetrate into the brain, and then we see the brain penetration by the decrease of the activity of the excitation by the AMPA injection. And this is reported here to have the comparison between BGG492 and AMP397. We have a similar percentage of inhibition in the brain after peripheral administration, demonstrating the penetration of the compound into the brain. Next slide. The in vivo epilepsy models are, of course, important because they allow us to do two things. First, really demonstrate that we don't just go into the brain and have some electrophysiological activity, but that we really act on the mechanism of epilepsy. And two, to try to understand what are the doses that we can predict to be the right dose for our clinical trials. And the first test that we use is the maximal electroshock seizure test, where we take mice, we expose them to a small current that is given to their brain via 2 frontal electrodes that we basically apply on the front of the brain and give a small discharge. And this small discharge as such doesn't cause epilepsy, it just causes a perturbation of the excitatory system in the brain locally, and this excitation spreads over the whole brain. It spreads over the whole brain in a way that is similar to generalized epileptic seizures in humans. And we see that the activity of this drug, BGG492, in mice and rats is very strong, with an ED50 of 7 milligram per kilo first hour. It stays for 2 hours, 3 hours, and then it starts decreasing in mice. In rats, it's a little faster. We see 10 milligrams per kilo, 8, maximum efficacy after 2 hours, and then it starts decreasing. It's an important model because it's also one that allows to measure tolerance, which is an issue encountered by many anti-epileptic drug candidates that work very well during the first day, and then a little less the second day, a little less the third day, and then they don't work anymore. And BGG492 actually worked as well after 10 days than after the first day, so that was very good. Next slide. The next slide shows a different model of epilepsy. You might have heard about absence epilepsy. It is an epilepsy form that touches children mostly, which doesn't cause as spectacular an epilepsy as the grand mal epilepsy that we hear about, where people really fall because they get convulsions and lose control. Absence epilepsy is simply a short duration epilepsy where the children lose contact with their environment. So they freeze for a few seconds. They just are not aware of their environment anymore. And this doesn't sound very dramatic, but in fact some children have this repeatedly every few minutes or every few hours. If it's every few hours, maybe the consequences are not too bad, but if it's very frequent, it prevents them from going to school, it prevents them from playing with other children because they just can't understand what they're being told. It's like having a sentence of which the middle of it is disappeared, and they don't have a normal behavior, so it's difficult for them to integrate in the society and play with other children. And fortunately, there is a strain of rats that has exactly the same phenotype. So they do also freeze, and we observe this just by looking at them. They run around in their cage, and all of a sudden they stop moving. And they stop moving for a few seconds, for maybe 1 minute at most, and then as if nothing had happened, they continue doing what they were doing, and they continue moving. And in fact, if you do an electroencephalogram of these rats, you see that during the period when they stop moving, their brain is going crazy. So you see the normal activity of the brain, and then all of a sudden, you see these very strong spikes, this very strong activation of the brain, which stops, and then the normal behavior comes back. This is exactly what happens in humans. So it was important for us to test in these rats the activity of our drug. And these animals have a number of bursts of about 10 to 20 per hour. It varies a little bit during the day, as you can see. So during the first day, we see a constant number, or actually a variable, but a sustained number of bursts per hour. And then after treatment, during the second day, we see that this comes down almost to zero, demonstrating the activity of our drug. It doesn't last very long because, as you know now, in rats, the elimination is very fast. But this doesn't matter. It shows us the efficacy or the predictive efficacy for absence epilepsy, which is really important for us. And the next slide is maybe even more important. It demonstrates the efficacy of this drug for two parameters. The first one is in a model where animals are given via an electrode small electric impulses in the brain, which causes a very mild epilepsy crisis. And then with time, this epilepsy crisis gets worse and worse every day. It gets stronger and stronger. And this is the mechanism of epileptogenesis, the increase of epileptic sensitivity that we see in humans. Remember Peter the Great? At the beginning, after his encephalitis, he had only very minor epileptic crisis, and then it got worse and worse with time. This is exactly what happens in this model. And of course, this is an extreme model because nobody has two electrodes in the brain discharging electric shocks every day. But we are very happy to see that BGG492 does actually decrease the severity of the seizures, the increase of the severity of the seizures in this model. It also showed an effect on seizure severity on animals that are what we call fully kindled, so animals that have reached the maximal severity of the epileptic crisis, where we see in this graph bar the first control day, second control day, and then vehicle treatment, no effect obviously. First control day, second control day, and small dose of BGG492, and a bigger dose of BGG492. It doesn't look like a very big effect. However, if you compare to MP397, and if you can click once, please, you see that the effect is about similar. And can you click, Alexei, please? Thank you. And this is the level that was predictive for the efficacy that we saw in the clinic. So we were very happy to see this efficacy, and we expect a clinical efficacy also in treatment-resistant patients with BGG492. Next slide, please. Of course, making an efficacious drug is important. We also need to make sure that it's safe, and they routinely go through a large number of toxicology and safety tests. BGG492 appears in all these tests as very safe. There's no cardiotoxicity, phototoxicity, genotoxicity, reproductoxicity, etc. I'm not going to go through the details of this, but we could confirm this safety margin in the next slide, please. In 4-week toxicology studies in rats and dogs, where we saw actually in rats absolutely no effect until the dose of 200 milligram per kilo, where there was some ataxia, which is already quite far away from the efficacious dose. So we were very confident that we could develop this drug without strong side effects. Ataxia is just a lack of coordination, and it results from the exaggerated inhibition of the AMPA receptor. So it's basically the good efficacy of the drug turning bad at very high doses. And in dogs, we don't have an ED15 in dogs, but we also saw a very good safety profile. Thank you. So next slide. Yeah. So it seems that BGG492 is a good drug. It looks like a very good drug actually, but does it follow the rules? And now we completed this table that we saw before with the results of course, the ideal guideline, CNQX, AMP397, which ultimately failed, and BGG492. BGG492 is not optimal. Yeah, it still has a total polar surface area which is very high, predicting a low oral bioavailability. But we have seen that this is actually not the case in our animal models. And the PK is a little high, which is also a predictor for suboptimal properties. But so far, it looks like BGG492 is really a good drug. And the message of all this is that, yes, we have rules, we have recommendations, but it's important to keep in mind that sometimes being a little off these recommendations is acceptable. Next slide. Summarizes, if you can click, yeah. Summarizes what I've presented today. We have identified the MP397 as a clinical candidate which proved the concept of AMPA antagonist in the clinic. This compound could not be developed further because it binds to bones and turns them orange. Click. please. Yeah, thank you. We had the BGG492, which was identified by chemical optimization based on this compromise between potency and oral bioavailability. This molecule became our clinical candidate and shows properties that are very similar to MP397, including a very broad spectrum of anti-epileptic activity and efficacy against epileptogenicity, which is a very important parameter. So this compound has the potential to treat pharmacoresistant patients. And it has entered clinical development in epilepsy, but also for migraine and tinnitus. So I don't want to go into the clinical part of this development, but I hope I could show you what are some of the issues that we have to grapple with as we go through the clinical development of a drug. And on the next slide, you will see that this is not my work I presented, but the work of many people. And I would like to leave this slide as a testimony to the interdisciplinarity of these efforts. Organic chemistry, as I mentioned at the beginning, is really the core of it, is the very beginning of it, but we need a lot of additional people contributing to drug development. And they are represented here for the preclinical part. And on the next slide, I'd like to leave you with a few pictures of Basel and Novartis in Basel, which is a very pleasant environment, especially when it's sunny like this. A very pleasant environment to work with, and which you are welcome to visit if you come see us in Basel one day. Thank you very much. Thank you, Alexei, for helping with the slides.
I'm a good switcher. So dear colleagues, we have some time for questions. Okay, so maybe one question and then we will have to go for lunch. But I just want to... Okay, sir.
Thank you. Thank you for the beautiful talk. One question, if it's not a secret information, on which stage of clinical trials is your drug candidate is now?
It is in phase 2, actually. If you're interested by the clinical study, they're all published on clinicaltrial.gov, so they're accessible. Yeah, phase 2, yeah.
So it seems like, unfortunately, we don't have time for any other questions, right?
That we have the possibility to discuss this presentation and lecture during the lunch because of time. Thank you very much for your beautiful presentation and lecture. As me, I pick up many interesting things.
Thank you.
From this lecture, thank you very much. If you like, ask some question. We can shift the time of the lunch.
Or maybe really, if you have a few questions, we can maybe spend 5 minutes on that. Or otherwise, I think we will have a poster session after lunch, right? So during the poster session, you're welcome to ask my colleague, Professor Carrera, and you can also talk to me. Actually, I want to tell you a bit more about Novartis, but we can discuss this in the private conversation if you like.
Thank you very much.
Okay, thank you.
Thank you.